posting to YouTube. So there's a biochemistry YouTube channel, and I think that's what I'm going to end up doing. So for those of you that are streamers, you can give me advice, but if it involves editing, it probably won't take your advice. So, but I've put up a couple things, and they seem to be good enough quality. People can hear them. They're usually me rambling at the beginning and rambling all the way through, I suppose, too. Lectures, lecture videos, I should say, on YouTube. And I'll put a link on the website if I haven't already. I've sent you already a link, I think, by email. And maybe the link's up on the website as well. We have lab tomorrow. What I'm going to do, I'll just print those labs and bring them in terms of directions. It's the pipetting lab. There's not going to be a pre-lab, so you don't really need to read ahead of time or prep for that. That won't always be the case. Questions on what we're doing in the class? Anything? Okay. Then last time what we had talked about, we talked about free energy. And at the end, we'd gotten to the point where I think I've just written the equation for calculating what I like to call real free energy. So real free energy or delta G. So real free energy, really it's just, people would just say free energy. There's free energy and there's standard free energy. And so I will try to say that from here on out. But what I mean by real is the free energy in the system you're looking at based on the concentrations of the things around it. Okay? Because that's real, it's important to know kind of what your driving force is for a reaction based on what's going on. And cells use these sorts of things to drive reactions. Cells will keep the concentrations of products of a reaction low so that it drives the reaction towards the products, right? That's a Le Chatelier's thing. So let's say we are, well, actually, let me give you the equation. So the equation is, the free energy is going to be the standard free energy plus a correction factor. Where Q here is the reaction quotient. And it's just like the equilibrium equation, although it isn't stuff at equilibrium, right? If it was at equilibrium, what would this be? Zero, right? So it isn't necessarily at equilibrium. It could be. And I'll show you how we use that in a bit. So let's say we had a reaction and when A plus B goes to C plus D. Well then, if that was the case, Q would be equal to the concentrations of the products over the concentrations of the reactants. Like that. Yes? Do you need to remember what?
No, they can't.
Oh, go move the lab codes. Could somebody else go and do that and pull it across? You guys are far more fit than I am and able to do that. Thank you. Thank you for asking. Just, is it on something? Just pull it out over there somewhere. I appreciate it. Okay, so then you would put the concentrations of those things at that time in the cell, and then you would have an idea of, in that particular instance, what you could calculate what the driving force was there. This is temperature in Kelvin. This is the ideal gas constant. And which one you use matters. You're going to use the one that makes your units all cancel out. Okay, and I'm not going to trick you and change it around. All right, so our ideal gas constant we're going to use in here is going to be, well, I'm not going to trick you, but depending on whether you use kilojoules here per mole, right? You could use kilojoules per mole, or whether you use joules per mole, will matter what you do with your ideal gas constant to make sure that cancels. So we're going to use 8.314 joules per mole Kelvin. Okay, that's the one we're going to always be concerned with. Yes.
Will we be given that on a test?
I will give you any constants on a test.
Okay.
I will never give you I'll never give you equations.
Okay.
Okay. All right. So let's go ahead and do a problem with this. We're going to be talking a lot about ATP in this class. And so in a muscle cell, the concentrations of ATP, ADP, and phosphate are the following. And I'm going to make up sort of, let's say it's an active muscle cell. I'm going to make up sort of reasonable concentrations. So let's say the ATP is 5.0 millimolar. The ADP is, let's say, 0.3 millimolar. And the phosphate is, I don't know, let's go ahead and we can make it 5.0 millimolar as well. Well, yeah, that seems fine. Okay. What is the free energy for the following reaction? And so we're in a muscle cell, so the reaction I would be running with that would be this. It would be ATP plus H2O goes to make ADP plus phosphate. So I forgot to put the concentration of water in there. Does that matter? It doesn't. Okay? The concentration of water is always built in. The concentration of the liquid or the solvent is built in. Remember that? If it's a liquid, if it's your solvent, or it's a solid, right? You don't worry about it. But if we look at these the rest of these things are going to be aqueous. I'm mostly never going to write these what the states are. But these are going to be aqueous. So why doesn't it, or why can you build this concentration into your free energy constants? What's the concentration of water in a cell, roughly? High. Does anybody know off the top of your head? It's about 55 molar. So approximately 55 molar, right? Okay, so I lied to you. There are some things in your body that are more concentrated than 1 molar. That's about it, okay? So you're made of mostly water. It's 55 molar, so when this reaction happens, the concentration of water doesn't change, right? You could have all the ATP turn into ADP, and this wouldn't change at all to the number of significant figures I have here. I'd have to add 3 more significant figures before it would matter. Does that make sense? Okay. So didn't include it, and that was okay. So now what we want to do is we want to apply our equation here, which is the free energy change is equal to the standard free energy change plus our T logarithm of Q. Oops. Let's go ahead and applying that, we can go just start plugging numbers in. So our standard free energy change, anybody for that reaction, right? The hydrolysis of ATP, anybody accidentally memorize it yet? Negative 30.5 kilojoules per mole, okay? Plus R, what R do I need to use now? I'm going to use the 8.314, but what do I need to do to it? I need to convert it, right, to kilojoules. So I need to divide it by 1,000. I'm just going to say that and do that. If you are never sure whether to multiply by 1,000 or divide by 1,000, what I recommend you do is you do a little bit of dimensional analysis or unit conversion for that. Actually, I will do it here just to show you what I would do. So if I wasn't sure, the way I would do this is I would write 8.314, right? Joules per mole Kelvin. And I want to convert that to kilojoules. Well, I know that if I have, see a joules on the top, if I have 1,000 joules, I have 1 kilojoule. And joules cancels with joules, and I'll have the right units now. And so that just did it for me. Okay, I recommend you do this unit cancellation stuff as much as you can. All right, if you're absolutely always just clicking things through your brain, you never need to do it for certain conversions. Don't start doing it. But if you make mistakes, do it. Okay, because this is rock solid. So then if I put that into my calculator, which I forgot. Oops, sorry, now I'm in kilojoules. Kilojoules per mole times Kelvin. Okay, that's what I get out. So I'm going to move that up here, 0.0008. I could have saved myself some pain if I'd used scientific notation here. Times the temperature. What's body temperature? Let's use. I didn't give you the temperature. I forgot. I'll remember on a quiz hopefully.
98.6 degrees Fahrenheit.
Okay, 98.6, and not Kelvin. Well, ask in Kelvin. Why do Kelvin? Or let's do in Celsius. What is it? 37, right? 37 degrees is your body temperature. So we'll do body temperature. 37 degrees Celsius plus, what do I do to make that into Kelvin? 273 equals 310 Kelvin, right? So go ahead, 310 Kelvin up here. Logarithm products over reactants. So the ADP concentration was, I think it was 5 millimolar. Okay. So I'm going to do this, right? Like that and that. I don't include water, right? So I'm going to have 2 things on top and 1 thing on the bottom. So if I had equal numbers of things on the top and equal things on the bottom, and they were in the same units, what could I do? I could cancel them and I wouldn't have to convert my units, right? I wouldn't need to worry about what my units were. However, I have not equal things in the top. So what do I need to do for my units? I had millimolar and more millimolar. I need to put them all in molar. If I don't put them in molar, I'm going to get the wrong answer, okay? Because everything doesn't cancel. Does that make sense? Okay? All right, so if what I said, though, wasn't like, oh, yeah, of course, just always put it in molar and you'll be safe, okay? You can always just convert everything to molar and you're safe, regardless. So that's going to be, I'm just going to do these things in my head. So I'm going to do 5.0, oh, that's a product. Whoops, sorry, that's a reactant. Well, it's a product too, okay? But doing them in order, this was ATP 5.0 times 10 to the negative 3 molar. This over here is, I'm going to have, it looks like I've got a 0. This was the ADP times 10 to the negative 3 molar. I'm going to do bad scientific notation there. And then 5 times 10 to the negative 3 molar. Okay? Does that make sense? All right, so now I can actually remember that. What I can't remember is I forgot to bring my calculator. So I'm going to go ahead and just scoot out to Google. Let's see if Google can handle this. So I'm going to do, it was 0.3 e to the negative 3 times 5 e to the negative 3 divided by 5, which was going to, oh gosh, it's easy, isn't it? Get up to the board, you lose like, I have 2 things that we're going to cancel. So it's just the 0.003. So when you get to the board, you lose about 20 IQ points. I made a similar mistake in my class earlier today. I just did something stupid that I could have done in my head. All right, so those cancel like that. So now it becomes, now I really need a calculator, or maybe Google can do that too. All right, let's try that. We're going to see what Google can do here. So what we want now is we want to take, it was 0.008314, right? Times, it was 310 Kelvin, times the natural logarithm. Let's see how good you are, Google. Are you really good? And then it was of 0.003, right?
One more zero.
One more zero? Oh, you're right. Yeah, because it wasn't 3 millimolar. It was, okay. Okay. We think so, maybe? And then what we have to do from that is we need to subtract, oh, no, we're adding, because we're adding we need to add negative, so we are going to subtract. No, that's fine. We're going to subtract 30.5, right? Okay? This is what you'd be doing in your calculator, right? You're not going to get to use Google in your exam. I will try to remember my calculator next time, although, you know, negative 51.4 kilojoules per mole. Okay? So we were just using Google to add these things up, and we ended up with negative 51.4 kilojoules per mole. Questions?
Wait for the negative 20. What did we do after that?
So we had the negative 20. This ended up being your negative 20, right?
Yeah.
So approximately negative 20. And then we added negative 30 to it. Okay? Yes?
Are we, with the for the purposes of this making example, are we ignoring stick figures?
We are mostly ignoring sig figs. So in this class, if you give me like one sig fig, I'm going to take off credit, or if you give me everything out of your calculator, I'm going to take out credit, but mostly I just care that you're plus or minus a couple. Okay? So we'll do that. Plus or minus 2 sig figs is okay. I don't want you spending time worrying about it.
Did anyone else get that value?
Yes.
Good question. So try it in your calculator again, or try writing out the parts individually and doing them. Okay. Any questions before I move on? All right, so you want to be able to use that equation. All right, so then what I'm going to do is we're going to talk about oxidation reduction. Because a lot of the reactions your body is doing, or any cell does, are oxidation reduction. And as an example of how this might be important, I'm going to show you this picture. Anybody know what condition this is? Jaundice. Okay. Anybody know what causes jaundice, or not the disease, but what is jaundice? What's going on?
Liver failure.
Okay, you got liver failure, but why yellow?
It's like building because the liver is connected to the gallbladder, it like causes buildup of stuff and it goes into your body and just kind of everywhere.
Okay, so there are a number of things that will cause this yellow color in your eyes. I'm going to give you a different example than what liver failure would do, but liver failure is one example. So jaundice is caused by the breakdown of hemoglobin. So the color. Okay, and one of the breakdown products of hemoglobin, in fact, it's a breakdown of the heme, is called bilirubin. All right, so we can come down below here, and I've got a picture of bilirubin. So this is, if you know what a heme looks like, and if you don't now, you will. But you don't need to know right now. This is the heme, essentially. that's been broken open, and it has that yellow color. So when we talk about the yellow color coming from breakdown of hemoglobin, most of the mass of hemoglobin isn't the heme. It's this other stuff. Yes?
Sorry, I didn't get the last bit of the. Can you just
Scroll up so you can get it? Sure. And remember, I'm always happy to do that. I want to work with what works for you guys. But I also post all these notes. Good? Okay. All right. So, well, why you got some different reasons why you might have the breakdown of hemoglobin. I want an example that involves oxidation and reduction. And there is a disease that is called glucose 6-phosphate dehydrogenase deficiency. Okay, this disease is interesting from all sorts of perspectives. It's the most common genetic disorder in the world. Okay, so it clocks in with 400,000 people have this, or 400 million people have this. Just a few zeros. Okay, and 30,000 people die from it a year. Now that's the whole world, and to put it in perspective, you've got about 40,000 deaths from flu every year in the US. Okay, so it's a lot of people, it's not a lot of people. Okay, and so what goes on is I'm not going to actually show you the part of the pathway involved in this right now. I may show it to you later. I may show it to you in a couple lectures, a lecture or two, or certainly we're going to talk about it in second semester when we're talking about metabolism. But what glucose 6-phosphate dehydrogenase deficiency, or actually what glucose 6-phosphate dehydrogenase does, is it makes something that is important for recharging the antioxidant system in your body. So glucose 6-phosphate dehydrogenase makes a product that is important for recharging your antioxidant system. Okay, and so what that means is your blood ends up being more oxidized than it might otherwise. It's more oxidizing. It's a more oxidizing environment. And it makes you vulnerable to all sorts of things. So this is an example of this, but you're going to have your cells. They're going to put Heinz bodies on them. They have bite kind of marks on it. They'll pop and blow up. All sorts of bad things are happening. And anytime you get hemoglobin breaking down and you can see it, the color, that's a bad thing, right? Regardless of the cause. In this case, this is the cause. And so all sorts of things, like they're oxidizing drugs like aspirin, these sulfonamides, which were kind of phased out, but with all the antibiotic resistance that's out there now, they're kind of going back to them. So I think I've been prescribed these, or maybe my daughter's been prescribed these. I'm trying to remember. But there's a number of drugs that do that. Faba beans. Anybody know what faba beans are? You use them to make falafel if you were in the Middle East. Okay, they're eaten more in the Middle East. We use probably garbanzo beans here. You've probably heard of doing that. But they're not like garbanzo beans necessarily. But they're very oxidizing. Okay, and this is actually such an effect that in ancient times, Pythagoras, who had a bunch of followers, wouldn't it be nice to be a mathematician that had followers? Okay, he had a bunch of followers, so he had some sort of Pythagorean cult, and he told his followers, forbid them from eating fava beans, because too many people died from eating fava beans. But they died not because the fava beans are necessarily bad for you, but they're bad for you if you have this disorder. Okay? So the various things, and infections also, there are infections that are oxidizing as well. So it's not that you don't have enough antioxidants to just kind of go along, but depending on what insults you have from your environment, you could be in trouble if you have this deficiency. Questions? All right, so that's an example of why oxidation and reduction are important. So let's go and talk about those. We're going to use two definitions in this class. First definition, we'll go ahead in order of the classes you took. For general chemistry. What was your definition for oxidation and reduction? What was it? Oil rig, right? Loss or gain of electrons. So we can talk about oil rig, right? So oxidation involves the loss of electrons. Reduction involves the gain of electrons. Okay? How about organic chemistry? Yeah.
And we based it off of the more bonds to oxygen is like oxidation, and then less bonds to oxygen or more bonds to carbon, I think is
not more bonds to carbon, but less bonds to oxygen. So let's go ahead to oxidation. So for oxidation, we'll go ahead and more bonds to carbon, or fewer bonds to hydrogen. Yes, I did mean oxygen. Thank you. So more bonds to oxygen. And then reduction. In reduction, you're going to have fewer bonds to oxygen. And more bonds to hydrogen. And which one we use will depend on what's going on. They're both easier in certain circumstances, or sometimes it's even impossible, at least from my perspective, to easily do it. So let's go ahead and look at a reaction. Now, if something's oxidized, what else must be going on? Something's going to be reduced, right? Okay, so that's something we want to keep in mind. So if we have, let's say we start with methane here. And what we can do is we can actually track on the atom basis for the most part, but we'll track with the heavy atoms at least. We can track what's being oxidized and what's being reduced. What's happening to these carbons as I go from the left to the right? They're being oxidized. We have more bonds to oxygen and fewer bonds to hydrogen. So this is actually, let's make oxidation red. I'll just do an ox. So if carbon's being oxidized, something must be getting reduced. What's getting reduced? Oxygen, right. Which I don't know if in organic they talk about that in that manner, but this is a reduction right here. And in fact, you know, you could think about this. This is, you, actually, I'm not going to say that. But this is super stable, but I don't know that it actually helps. They're both super stable. O2 is reasonably stable. H2O is super stable. It's actually, when you think about, we're not going to do the electron transport chain this semester, but oxygen or water is kind of the final, oxygen's the final electron acceptor to make water, which tells you it's really stable. Okay, we could look at this. Another common, at the end of glycolysis, you make pyruvate. This is the structure of pyruvate. What happens if you're working out really hard? You make lactic acid, right. Your pyruvate gets turned into lactate. And your body's doing this so that it can just keep running glycolysis a little bit longer. We'll talk about that more next semester. All these great teasers for next semester. So it's going to use this high energy molecule to make that happen. So there's our reaction there, right? Pyruvate gets made to lactate, and NADH gets turned into NAD+. What is happening to my pyruvate? It's the one of the, this is pyruvate right here. It gets reduced. Why did you say that?
Because there's a hydrogen that goes to the oxygen.
Yeah, we're adding hydrogens, right? How many hydrogens did we add?
1.
2. Yes, 2. You added 1 here, and you added 1 there, right? Right, so when you see this sort of thing, you're actually adding, you know how you added hydrogens across the double bond in organic? You're actually adding hydrogens across the double bond. It just doesn't look the same as maybe what you're used to. What's being oxidized? This is being oxidized, right? Okay, and you know, it's just an abbreviation, but I do see it's losing a hydrogen, so that would be one of my clues there. All right, but if you're given a reaction and you can easily tell what's being oxidized or what's being reduced, then you already know the other one. So do the easiest one. Okay, I had a couple more I wanted to do here. Let's go ahead. So in hemoglobin, it's really important that your iron be kept in the +2 state. But sometimes it goes to the +3 state, which is bad. Okay? What's good? This is actually just a half reaction here. I'm not giving you the other part. So what is this? If I go from +2 iron to +3 iron, that's oxidizing, right? It loses electrons. You're losing minuses, right? I have to take a minus away to get +3. So electrons are lost. So it's oxidation. Questions? Okay. That is it for that stuff. I'm going to skip past this, and what we're going to do now is start into talking about natural selection. So I talk about natural selection for a day, and the reason why I do that is, you know, I talk about in chemistry, you can explain 90% of everything using charges, right? Well, in biology, you can explain why something is the way it is using natural selection, right? Why is that way? Well, there's natural selection there. Then you might be able to come up with an explanation that better explains why with that. So what we've got here, so natural selection is the differential survival and reproduction, otherwise fitness of individuals that differ in phenotype. Okay, so that's a really complex definition. It is a good solid definition, but it's complex. Let's break that down a little bit. First of all, we've got phenotype here. Okay, what is phenotype?
Characteristics, like the way you look.
So it's a characteristic. It's characteristic how, whoops, how something looks. Okay, so it could be your eye color is a phenotype. It could be, you know, how susceptible you are to oxidants. Oops, I'm not using spell check. This could be awful. Susceptible. How vulnerable? You are to oxidants. Okay, then I'm going to do an etc. There are lots of examples of this, but we'll explain these two. So when we talk about eye color, let's see if I can get my thing back. There we go. When we talk about eye color, so both my kids have blue eyes. I have brown eyes. My wife has blue eyes. My mom has brown eyes, and my dad has blue eyes. Okay, so I told my dad that the kids have his eyes. How can I do that? You guys remember how this works? Oh, I'm gonna erase this great linear algebra up here. This feels like wrong, but it's gonna happen this afternoon when they have lab anyway, right? So do you guys remember how the eye stuff works? Blue eyes work? You probably don't all. So somebody explain this to me.
It's a recessive gene. So
Which one is?
the blue eyes.
Blue eyes is, right. So we're gonna do, this is brown, right? This is not blue, but I'll blue with green. Okay, since that's what I have up here. All right, so what we have is somebody who has a brown-eyed gene and a blue-eyed gene. They're going to have, whoops, brown eyes. Because the blue is recessive. For somebody to have blue eyes, they have to have a blue-eyed gene and a blue-eyed gene. And that's going to give them blue eyes. Right? Okay, and so I said my mom has brown eyes, my dad has blue eyes. He could have only given me a blue gene, right? My mom might have a blue gene, but she didn't give it to me because my eyes are brown. So I know that, so based on that, based on my dad having blue eyes, I know that this is what I call my genotype. So Dr. Johan, my phenotype is, or my genotype is blue, right? And my eye color is brown eyes. All right, so then we have Dr. Sarisky. And her eyes are blue, so we know she has a blue gene, or two blue genes. How do you do this? We've got two kids. They both have blue eyes, right? So what that means is they got this gene from me and one of those two from my wife. So both my kids have blue eyes, but this gene way back here, this gene had to come from my dad because he has blue eyes as well. That make sense? Okay. So it's fun. All right. But the point of that discussion related to what we're talking about here is those phenotypes come from genotypes. So back in the you can see a color of somebody's eyes, but the eyes come back from derived from their DNA, from what's going on with their DNA in terms of these genes. So phenotypes, well, not genotypes, I should say. Okay, a genotype is it's the genes that determine the phenotype. In other words, it's the DNA. So genes are, well, I'll define them in a second. Okay, and so in terms of genes are sequences of DNA that produce a functional product. And I should say that contain the information for a product. I'm not even going to make it functional, okay, because it doesn't need to be. We could argue whether a cancer gene is functional or not, right? We could argue all sorts of things. Does that make sense? Do people have questions about that? About what a gene is? Okay, a genotype, a phenotype? All right. So what natural selection is about then is natural selection comes back to what DNA does that organism have? Okay? And so let's take a look at this example. This is an example, a classic example of natural selection. Some of you may have heard of it before. Anybody want to talk about what this one is? Yeah.
They're moths.
Yeah, they're moths.
And they have two different phenotypes.
Yes.
So there's a darker one and there's a lighter one.
Right. So here.
And they have a predator
Uh-huh.
that can easily see the darker ones.
Right.
On the birch tree.
Right.
And so they were getting eaten.
Right.
And the lighter colored ones were thriving and reproducing.
Right.
So the black ones were basically just funneled out.
They were getting funneled out, and then something happened. So this was happening in England. Perfect so far, right? This was happening in England, right? So it was better to be this kind of moth because there was birds were eating this moth because it was easier to see on birch trees. All right. So, but then what happened in England?
Industrial revolution must be down the birch trees, and they get more advantageous to be better for moths.
Right. So the Industrial Revolution happened, and you got a bunch of soot on the trees, and now all the trees were black, and now it was better to be this moth. And so it used to be that 90% of the moths look like this, and 10% look like this. I might not be exaggerating enough, actually. And then suddenly, that Industrial Revolution comes, and all the birch trees are black, and now 90% of the moths look like this, and 10% of the moths look like that. Does that make sense? Because the predator, it depends on what's easier for the predator to get to. Okay? So we're going to use natural selection for that. And so the explanation would be the phenotype of, let's say in this situation, before the Industrial Revolution, the phenotype of having the lighter color allowed you to escape predators easier, right? Made you more likely to escape predators and have children and pass the genes on to your children that cause this color. Does that make sense? Okay. So I'm going to ask you a number of different natural selection questions using that reasoning, but I want to kind of give you a template because a natural selection question has to have certain things in its answer. All right. So a full credit natural selection answer will have the following. It's going to have a description of the trait that's being selected. And I'm going to say being selected for or against. Okay, so that depends on how the question is phrased. It's going to have, and here's the most important part, this is the meat of it, a detailed description of how that trait is selected. Okay, and then finally, it's going to have a description of the consequence of that selection. Let's go ahead and fill this in for our example up above. I would like blue text, please. Let's see how easy this is. So the trait that's being selected, let's say 4 up here, okay, in the case of the picture we have. So before the Industrial Revolution, the light colored phenotype was being selected. And I'll say 4, right? Better to have the light colored phenotype. Detailed description of how that trait is being selected. Okay, so the detailed description of how that's being selected here is that birds could see the dark colored, actually let's make it very specific to that phenotype. Birds couldn't see the light colored moths on birch trees as well as the dark colored moths. Thus, the light colored moths were more likely to escape being eaten. All right, and the consequence of that, which may often be the exact same thing you write. There may be some problems where it doesn't quite fit, but the consequence of that selection is one, 2, 1. Lovely. Anyway, 1, 2, 3. The light color moths were more likely to survive and have offspring. And pass on their light colored genes. Questions on what an answer should look like? I'll probably even, I don't promise, but usually I even scaffold it like that for you in the question. Yes.
Do you mind to say how it changes?
How?
Like, 'cause the Industrial Revolution, it switched
Yeah, but that wouldn't, that's not real. So I don't have the question here, right? Let me add a question on top of it. So I formatted the first one like the question was. It's a good question you're asking because this will make it more clear. Okay, so before the Industrial Revolution, birch trees were light colored in England, and light colored moths were far more abundant than dark colored moths. Okay, explain this observation using natural selection. See if I can figure out how to, no, move. I'm trying to figure out how to separate these. There. How's that? Questions? Okay. So that would be an example of a question. This would be how you would answer the question. Except that one of my sentences moved. And now it's all ones. I'm not going to fix the all ones, but I'm going to move the sentence to the right place. Okay, so imagine 2, 3. All right. Any other questions on that? Okay. Well, let's. I'm a little ahead of my notes here. I'm not going to let you go early because it's way too early in the semester for that. I'm not tired yet. So why don't we try a practice problem on some of the math stuff we talked about. Actually, let me go ahead and set up another one of those. Well, we could do creatine. So let's do that. Creatine phosphate is used to regenerate ATP by the following reaction. Okay, concentrations are given, and assume body temperature, which is 310 Kelvin. Calculate the free energy for this reaction. Okay, so what we've got is our reaction is going to be, it's going to be the following. We're going to have ADP, right, combines with creatine phosphate or reacts with creatine phosphate to make ATP, which is going to allow you to use your muscles some more, plus creatine. All right, The standard free energy for this reaction, we said was, we calculated that yesterday, right? Or last lecture. That was negative 14.5 kilojoules per mole, I think. Okay, let's say our ADP concentration is what it was before, which was 3.0 times 10 to the negative fourth molar. Oh, I just put it in molar. We don't have much time anyway. Our creatine phosphate is going to be 30, so let's say 3.0 times 10 to the negative 2 molar. Our ATP is going to be about 5 millimolar, so 5.0 times 10 to the negative 3 molar. And our creatine is going to be really low when we start this, so let's call it 1 times 10 to the negative 4 molar. So go ahead and work that. And remember that your free energy, your R, is 8.314 joules per mole Kelvin. Anybody need me to write the equation down? Yes.
So are any of them like the water in the last, like, we use all the stuff in the ocean?
What do you think? Just what? Are any of them a solvent?
No.
No.
Okay.
So, but I'm not gonna, like I said, I'm not usually gonna write these. The only real solvent you're ever gonna encounter in biochemistry is water, right? We're not, I don't even think we do anything in lab that doesn't involve water, but no, we use some acid. We do a couple TLC plates, so you know, and they do some stuff for that. Anybody get it? What'd you get?
Negative 21.9.
Negative 21.9 kilojoules per mole. Anybody else get that? Okay, so we got 2 people who got it. So if you want to write that down as your answer for now, I'm going to go back to my office and I'll work it, and so I'll have the work there, and then I'll check my answer and see if I got it right. All right, I'm going to